Good morning, children. Welcome to our next class of biobotany. In the previous classes, we started a new lesson, lesson 16, dealing with plant hormones. So, in that we studied there are 5 types of plant hormones found in the plant. Some of them, they are the plant promoters, growth promoters, and some of them are growth inhibitors. So we studied the 5 plant hormones. They are auxins, cytokinins, gibberellins, abscisic acid, and ethylene. So among this, we studied auxins, cytokinins, and gibberellins. They are growth promoters, whereas abscisic acid and ethylene, they are growth inhibitors. Growth promoters means they help in the growth of the plant, and inhibitors means those which stops the growth of the plant parts. So in the previous class, we studied about 2 plant hormones in detail, that is auxins and cytokinins. So today we are going to study the remaining 3 plant hormones. They are gibberellins, abscisic acid, and ethylene. So first let's study about gibberellins. Now among all the plant hormones, all the 5 plant hormones, the most abundantly found plant hormone in the plant is gibberellins. Okay, so gibberellins, they are the most abundantly means they are found in large quantity. Gibberellins, they are found in large quantity in the plant. Kurosawa in 1926, he observed Bacne disease or foolish seedling disease in rice crops. So this is related to the discovery of this gibberellins. Kurosawa, he found that in the rice field, when all the rice seedlings were planted at the same time, some of the rice plants, they show extraordinary growth, or some of the rice plants, they were taller than the other rice plants. So here, when he made an observation in these tall plants, they found that in the internodal region of these plants. Now, what is the internodal region? The region between the 2 nodes. Usually, you can identify the nodes. From the nodes only the leaves arises or the branches arises. Here, the region between the 2 nodes, that is called as the internodes. So here he found that in these rice plants, these internodes, they were longer than the other rice plants which showed the normal growth. Okay, so that means the internodal elongation was found here, and this was found because and he made an observation that this elongation in the internode was because of a fungus called Gibberella fugicori, and he found that this Gibberella fugicori, they secreted some active substances in this plant, and that was called as gibberellin. This gibberellin helped in the elongation of the plants. Okay, so this disease was called as Bacne disease, or this disease in the plant was called as the foolish seedling disease. Okay, so here we have studied about the history of the gibberellins, how it was discovered. Now, what is the role of this gibberellins, or what is the physiological effects of gibberellins? What is the effect of the gibberellin in the growth of the plant that we are going to study here? Application of gibberellins on the plants stimulate extraordinary elongation of the internode. So here in the plant, if we apply this gibberellins, what will happen? The plant will show extraordinary elongation of the internodal region. So plants in which we can find this extraordinary elongation is corn and pea. Next important effect of gibberellin is a bolting effect. Now here, first you can see in this diagram, now 2 plants are there. The first plant, it is showing rosette leaves. Now what is this rosette plants? The plants which do not show growth in the stem. Okay, the growth of the stem is inhibited. So what will happen? All the leaves, they will grow very close to each other, and they will give a rose appearance. So such type of plants, they are called as the rosette plants. So here, when the plant is not elongated, the height of the plant is not increasing, all the leaves, they will grow very close to each other. This type of plants, this short height plants, they are called as the rosette plants. Okay, so here when these rosette plants, they are treated with this gibberellins, the shoot of these plants, they show extraordinary elongation. Okay, so shoot, they start growing a normal growth, and they will the leaves will also spread out. Okay, And after this only you can find, when the shoots they attain a normal growth only, you can find the flowering in this plant. So this is called as bolting. So treatment of the rosette plants. So rosette plants, I told you, they are the plants which do not show the growth increase in the height of the plant or the size of the plant. As a result, all the leaves they grow very close to each other and they give a rose petal like appearance. So here treatment of this rosette plants with gibberellin induces means causes sudden shoot elongation followed by flowering. Now here previously it was an abnormal condition. The plant was not showing the normal growth. As a result, there was no flowering. Now when this plant is treated with the gibberellins, the plants they show the normal growth and they produce the flowering. This is called as bolting. So first physiological effect of gibberellin we study, it causes the extraordinary elongation of the internodes. Second we studied, it induces the shoot elongation in rosette plants followed by flowering. Next we are going to study about the next physiological effects of gibberellin. They are gibberellin, they produce the production of male flower in monoecious plants. Now what are these monoecious plants? Now monoecious plants, they are the plants in which the male flower and the female flower they grow separately in the plant. Okay, so male flower they grow separately in the plant, female flower they grow separately but in the same plant. Then it is called as monoecious plants. So here the plants, if they are treated with the gibberellins, there is more production of the male flowers in the plant. Okay, so monoecious plant, I told you the monoecious plants are the plants in which the male flower and the female flower they grow separately but in the same plant. So gibberellin promotes the production of male flower in monoecious plants. So example of this monoecious plants is cucurbits. Next physiological effect of gibberellin is gibberellin breaks the dormancy of potato tubers. Dormancy means a condition when the plant is not in an active state. It is alive but it is not in an active state. Okay, so here as we know, from a potato tuber we can raise a new plant. So here when these potato tubers they are treated with the gibberellin, they break the dormancy state. Okay, that is the inactive state. They will break that inactive state and they will produce the buds and they can develop into the new plant. So gibberellins they break the dormancy of potato tubers. And the next physiological effects of gibberellins are efficient than auxins in inducing the formation of seedless fruits, that is the parthenocarpic fruits. Already we have studied, when we are studying about the auxin, we studied that is the fruit formation takes place only by the act of fertilization, but some fruits can be made to develop without the act of fertilization. That is, any part of the flower can be developed into fruit without the act of fertilization. That is called as parthenocarpic fruits, and these fruits they are the seedless fruits. Okay, so here we have studied when auxins are when we apply the auxins we can develop the parthenocarpic fruit. But here when it is a comparison is done between the gibberellins and the auxins, gibberellins are more efficient. Okay, gibberellin can promote more parthenocarpic fruits compared to the auxins. So here the example for this is in the tomato plant. So more number of tomatoes can be obtained in the tomato plant by the treatment of gibberellins. Okay, so these are the physiological effects of gibberellins we have studied. So first physiological effect we studied, it causes elongation of the internode. Second we studied it causes the sudden shoot elongation in the rosette plants followed by flowering, and this process is called as bolting. Next we studied it promotes the production of the male flower in. Next we studied it can break the dormancy of potato tuber. And the last point we studied gibberellins can also produce parthenocarpic fruits. Okay, so these are the physiological effects of the gibberellins. Okay, next we are going to study the next plant hormone, that is abscisic acid. Now abscisic acid is also called as ABA. Okay, so abscisic acid is a growth inhibitor. Growth inhibitor already we studied. They are what? The plant hormones which can inhibit, which stops the growth of the plant. How they are crossing the stop? Which regulates the abscission. Abscission we have studied. It is the separation of the leaves, flowers and fruits from the plant. This is called as abscission. Okay, so it regulates abscission. It controls the abscission region in the plant. And dormancy. Dormancy also again we have studied. What is the dormancy? A condition where the plant is alive but it is in an inactive state. It increases the tolerance of plants to various kinds of stress. Okay, so as we know, what are the stress a plant can face? Soil stress. The condition of the soil is a factor of stress for the plant. Next, availability of the water. Okay, if there is more water is available or the less water is available for the plant, that is also a condition of stress to the plant. Next, you can say the temperature, the heat. These are the various factors which are also the stress causing for the plants. So this abscisic acid, it is said to be a stress hormone. It increases the tolerance of the plants to various kinds of stress. It helps the plant to withstand, to face the stress condition. So this is abscisic acid is also called as a stress hormone. Now it is found in the chloroplast of the plants. So abscisic acid, where it is found? It is found in the chloroplast. So here what are the information we have studied about abscisic acid? It is a growth inhibitor. It regulates abscission and dormancy. It increases the tolerance of the plant to various kinds of stress. So it is called as a stress hormone. And this abscisic acid, it is found in the chloroplast of the plants. Next, what is the physiological effects of this abscisic acid we are going to study here? So first, physiological effect, ABA promotes the process of abscission. Abscission we have studied. It is what? The process where the separation of the mature leaves, flowers and fruits takes place. Okay, the mature leaves and flowers and fruits, they are separated from the plant by the formation of an abscission zone. Abscission zone is what's a block, you can understand as a blockage where the vascular tissues are blocked, so the particular plant parts will not receive the food and the water. As a result, they start dying there. Okay, so ABA promotes the process of abscission. Next is during water stress and drought condition. So when there is lack of water or in the drought condition, ABA causes the closure of stomata. As we know, what are stomata? They are the small pores found in the epidermis through which exchange of gases and loss of water takes place. Now already the plant is growing in a condition where there is lack of water, in a drought condition. So if the stomata is open, what will happen? The plant will lose the water and the plant may undergo drying there, wilting will take place. So here the abscisic acid, what it is doing? It is causing the closure of the stomata so that the plant can retain the water. So here the next physiological effect we have studied, role of abscisic acid we have studied. It causes the closure of stomata during the drought condition. Next physiological effect of abscisic acid is it promotes senescence. Is what? It is a time period from the maturation to its death. Okay, or we can also understand as the plant matures, the cells start losing their function. For example, when a leaf is maturing, what will happen? How you can identify the leaf is maturing? It will start losing its green color, and the green color leaf will slowly start turning into yellowish color, then start into brown color, and finally it dry up. Here, so ABA promotes senescence in leaves by causing the loss of chlorophyll. Okay, so when the mature leaf, the loss of chlorophyll for the who is helping there? This ABA is causing helping there. Next, ABA induces bud dormancy towards the approach of winter in trees like birch. So here, what is happening? Now, as the plant, there is a in the winter condition where the plants they cannot withstand the extreme cold. So this abscisic acid can cause the dormancy of the bud. Okay, dormancy of the bud to withstand the winter season. Next, ABA is a powerful inhibitor of lateral bud growth in tomato. So lateral bud we have studied. Two type of buds we have. One bud which is in the tip of the plant, that is called the apical bud, and the other bud which grows in the side of the plant, they are called as the lateral bud. So this ABA, it is a powerful inhibitor of growth of the lateral bud. It stops the growth of the lateral bud, specifically in the tomato plants. So here the role of abscisic acid, as we have studied, first one what we have studied, it promotes the process of abscission. Second we studied, it causes the closure of the stomata in drought condition. Third we have studied, it promotes senescence in leaves by causing the loss of chlorophyll. Next, we have studied it induces the bud dormancy in the approach of winter. Next, it is powerful inhibitor of lateral bud growth in tomato. Next, we are going to study the last plant hormone in our syllabus. We are going to study is the ethylene. Now, among all the 5 plant hormones, ethylene is a gaseous plant hormone. Okay, so which is the gas? It is ethylene. Ethylene is a gaseous plant hormone and it is also a growth inhibitor. So we have studied 2 plant hormones which are growth inhibitors. What are those? Abscisic acid and ethylene. They are growth inhibitors. They inhibit the growth of the plant. Now this ethylene, the function of ethylene, the major function of ethylene is it causes the maturation and ripening of fruits. Okay, the maturation of the fruits and ripening of the fruits is controlled by this ethylene. So ethylene is a gaseous plant hormone and is responsible for maturation and ripening of fruits. Maximum synthesis of ethylene occurs during ripening of fruits like apples, bananas and melons. So here the ethylene is secreted in abundance at the time of ripening. Okay, in apples, bananas and melons. Now what is the role of this ethylene we are going to study here? Physiological effects of ethylene. First physiological effect, it promotes the ripening of fruits. Okay, so as we know, tomatoes when they ripen up, so when the unripe tomatoes, they are green in color. When the tomatoes ripe, so what will happen? It turns into red color. So here this promotes the ripening of the fruit is controlled by ethylene. So in the plants like tomato, apple, mango, banana, etc. Next, ethylene inhibits the elongation of the stem and roots in dicots. So next function of this ethylene is it will prevent the elongation, increase in size of the that is increase in the length of the plant shoot as well as in the roots in dicot plants. Next, ethylene hastens means fastens or speeds up. Ethylene hastens the senescence of leaves and flowers. Senescence already we studied. It is the as the plant matures, the cells they start losing their functions. Okay, so here again ethylene what they do? They speed up the process of senescence of leaves and flowers. Next function of ethylene is ethylene stimulates formation of abscission zone in leaves, flowers and fruit. This leads to premature shedding. Okay, so ethylene also what it does, it causes the formation of abscission zone in the leaves, flowers and fruits. As a result, what will happen? Premature shedding of the leaves or flowers or the fruits takes place. You can see sometimes small fruits, they before they mature itself they shed down. So here what is happening again, the formation of the ethylene is causing that. It may be a factor along with other factors. This is also a factor for the premature shedding of the leaves, flowers and fruits. Next is what? It breaks the dormancy of buds, seeds and storage organs. So here again, as we have studied, dormancy means a condition where the plant part is alive but it is in an inactive condition. So here the ethylene can be used to break the dormancy in the buds, seeds and storage organs. So what are the plant parts which can be stored in, then the dormant condition can be broken by can be stopped. Dormant condition can be stopped by using this ethylene. So these are the physiological effects of ethylene. So first one we studied, it is a causes the ripening in fruits. Next we studied it inhibits the elongation of the root and stem in dicot plants. Next it speeds up the senescence of leaves and flowers. Next it stimulates the formation of the abscission zone in leaves, flowers and fruits, and this causes the premature, before maturing of the leaves, flowers and fruits they will separate from the plant. That is called as premature shedding. Next, ethylene breaks the dormancy of the buds, roots and storage organs. So the stored seeds, their dormancy can be broken by using the ethylene. So with this children, we have completed all the 5 type of plant hormones. The next part of the lesson is dealing with the hormones which is secreted in the human body or in the human. Okay, so here that will be taught by a zoology miss. Thank you children.